Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, welcome back to the deep dive. Let's start with a thought experiment. What if the real measure of how you're aging, what if it wasn't just your age in years, your chronological age, but instead it was this kind of dynamic molecular report card?
Yeah.
Something you could get from, say, a simple blood test.
That's really the holy grail in a lot of aging research, isn't it? Moving beyond just counting years to actually measuring biological function, resilience, right down at the molecular level.
And that's exactly what we're digging into today. We're looking at some pretty groundbreaking research that combines advanced analysis of these tiny molecules in your blood metabolites, they call it metabolomics, with some really cutting-edge machine learning.
Right. And their goal was specific. Can we identify the unique molecular signature of what they're calling active aging?
We're basing this heavily on a new scientific paper. It just came out in MPJ Systems Biology and Applications alongside some health news summaries. It analyzes data from, I think, 263 samples from older adults.
That's correct. And it wasn't just a broad look. They were really trying to translate physical fitness, how well someone moves and functions, into the language of biochemistry.
So our mission today is to unpack that translation. We're going to look at how the researchers actually quantified physical fitness, how they condensed it down, then how AI could basically read the blood results with like 91% accuracy to predict that fitness.
And crucially, how they pinpointed one dominant biomarker, a molecule that seems to act as this measurable link between being physically active and potentially keeping your brain healthy long term.
Yeah, because we all sort of know exercise is good for you.
Right.
Right. That's the standard advice. Protects against disease, helps mobility.
That's a long-standing consensus, yes. But the big unanswered question this research really tackles is, can we actually point to specific molecules in the blood that show us quantitatively the benefits of an active lifestyle, especially in older individuals?
So moving past just exercise is good to exercise changes this molecule through this pathway, and that is why you're healthier.
Precisely. The aim here is decoding the exact molecular gears, the processes that turn physical activity into healthier aging that we can actually measure.
Okay, so let's start with the people in the study. The analysis used 263 blood plasma samples. These came from elderly folks living in retirement homes in Vienna.
And this is a key group, right? They're generally older, average age above typical life expectancy, mostly women in this case. And they're in a setting where aging and inactivity are, very real concerns.
And there's an important point about the study design, too. Originally, these participants were part of a different study with different groups. Some did resistance training, some had supplements, some did cognitive training.
Right. But for this specific analysis, the one we're focusing on, the researchers deliberately pooled all the data together. They weren't trying to compare the effects of, say, resistance training versus cognitive training.
Okay. So the goal wasn't which exercise is best.
Right.
It was simpler. Just look at everyone and find the basic relationship between their overall physical performance, however they got there, and what their blood chemistry looked like.
Exactly. They wanted to find the universal link between general fitness and the metabolic profile.
So to do that, they needed a solid way to measure general fitness. They couldn't just, pick one thing like walking speed.
No, because physical function, especially as we age, is complex. It's strength, it's endurance, it's how quickly you can get up from a chair, it's balance. You need to capture all of that.
So how do you roll all those different things, walking distance, chair rise tests, hand grip strength, balance tests, into one single meaningful score?
That's the challenge. You can't just average them. It doesn't really work statistically. So they used a pretty sophisticated technique called canonical correlation analysis, or CCA.
CCA, okay.
Yeah.
What does that actually do?
Think of it like a smart mathematical filter. Instead of just adding up the scores, CCA looks for the hidden connection, the underlying factor that best explains the correlation between all those different physical tests. It finds the shared element.
So it's identifying the common thread that links walking ability, strength, and balance together.
Precisely. It creates the most statistically sound single score that represents that overall physical capability. They called this the Body Activity Index, or BAI.
The BAI. Okay, that makes sense. A synthesized score for fitness.
Yes. And when they calculated this BAI, a really crucial insight popped out straight away. The single biggest contributor to that overall BAI score was walking distance.
That's interesting. So in this group of older adults, endurance, just the ability to walk a certain distance, seems to be the most dominant factor defining how active they are in this aging index.
It suggests that, yes, sustained mobility and endurance capacity were highly significant for their overall physical status as measured by the DAI.
Okay, so the BAI is the physical side of the equation. What about the molecular side?
Right. Independently, they created another index. This one was based purely on the blood tests. They measured the concentration of 35 specific small molecule metabolites and derived a metabolomics index from those.
Let's just quickly clarify metabolites again. We're
talking about the small end products of all the biochemical reactions happening in your body constantly. Things like sugars, amino acids, organic acids, basically the chemical fingerprint of your cells working. Eating, breathing, exercising, it all produces metabolites.
And they picked these 35 specifically.
Yes. They were chosen because they represent core pathways, energy production, amino acid metabolism that are known to be influenced by physical activity. So it wasn't a random selection.
Got it. So we have the physical score, the BAI, based on movement tests.
Yes.
And we have the molecular score, the metabolomics index, based on blood chemistry. The big question, do they match up?
They matched up incredibly well. The correlation between these two completely independent indices, the BAI and the metabolomics index, was extremely high. The Pearson correlation coefficient was 0.85.
Okay, 0.85. For people not steeped in stats, how significant is that in, messy human biology?
It's monumental, honestly. In biological systems where so many things vary, diet, genetics, mood, environment, getting a correlation that high is considered exceptionally strong, almost a perfect alignment.
Wow.
And the statistical probability, the p-value, was tiny, meaning it's extremely unlikely this happened by chance. It's a very robust finding.
So that's a massive validation right there. It means that this molecular signature in the blood really does accurately reflect the overall physical fitness measured by those traditional tests. Your blood chemistry mirrors how well you function physically.
Okay, so they've established this strong link. The blood reflects the body's fitness. But now the detective work really begins, right? This is where the smart blood idea comes in. Which specific molecules out of those 35 are actually driving this connection?
Exactly. Correlation is great. It tells you there is a relationship, but it doesn't tell you which components are most important or how predictive they are. And biological relationships are often messy. They're not simple, straight lines. So you need something more powerful than just looking at correlations.
Right. You need tools that can handle complexity, nonlinear relationships between fitness and these metabolite levels. And that's where machine learning, or AI, comes into play.
Let's unpack the AI approach they used, because it sounds like they were very thorough. They used 5 different machine learning models. Why so many?
It's all about robustness. Making sure the findings aren't just an artifact of one particular algorithm. They used a range from a relatively simple statistical model, a generalized linear model, kind of a baseline, up to much more complex ones.
Okay.
Like boosted decision trees, algorithms called gradient boosting machine, GBM, and XGBoost, which are very powerful for this kind of data. And even a deep learning approach, an autoencoder network. Using this variety helps ensure that if there's a signal there, one of these methods will likely pick it up reliably, regardless of how complex the pattern is.
And they didn't just train these models once, right? They used something called repeated double cross-validation. Sounds complicated. Can you break that down?
Yeah, it sounds complex, but the idea is simple. You want to be absolutely sure your model works on new data, not just the data it was trained on. That's called avoiding overfitting.
Like memorizing the answers for one test, but failing a different one.
Exactly like that.
Yeah.
So repeated double cross-validation is like simulating that real-world testing over and over. They did it 30 times. Each time, they split the data, trained the model on one part, and then tested its predictive power on a completely separate part it had never seen before. It's a very rigorous way to ensure the model's performance is genuine and reliable.
Okay, so it guarantees the model isn't just good at predicting fitness for these specific Viennese participants, but that it likely generalizes. So what did they find? How well could the AI predict fitness just from the blood?
The results were really quite striking, especially the boosting methods, XGBoost and GBM. They achieved extremely high accuracy. They could distinguish between the participants classified as active versus less active correctly over 91% of the time, based only on the molecular data.
Wow, 91% accuracy from a blood test.
Yes. Technically, the AUC, or area under the curve, was greater than 0.91. In clinical diagnostics, anything over zero point 0.9 is generally considered excellent predictive power. It strongly confirms that the molecular data contains enough information to accurately predict fitness level.
Okay, so the proof of concept is there. The blood can predict fitness with high accuracy. But the million-dollar question is, which metabolites were making the prediction? Which ones were the key players?
That's what the models revealed. Across all 5 different algorithms, which is important for consistency, a core group of 8 metabolites consistently emerged as the most important predictors of activity level.
8 key molecules. Do we know which ones?
We do. They were aspartate, proline, fructose, malic acid, pyruvate, valine citrate, and ornithine.
Okay, that's a focus list. That's the molecular fingerprint of active aging, at least in this study. Was there one that stood out more than others?
Oh, absolutely. While all 8 were important, one molecule was head and shoulders above the rest in terms of its predictive weight. That was aspartate.
Aspartate, an amino acid, right?
Yes. And it wasn't just slightly more important. The analysis showed aspartate confirmed its role as the truly dominant molecular marker. In the best performing model, the XGBoost classifier, aspartate alone accounted for over 90% of the variable importance.
90%. Just from aspartate. How is that possible? How can one single molecule carry so much weight in predicting something as broad as physical fitness?
Well, that's the crux of it, isn't it? It immediately tells you that aspartate metabolism must be incredibly central to the biological processes that define physical activity and fitness in this context.
So what does aspartate actually do in the body that makes it so important?
It has a couple of really critical roles, especially relevant to exercise. First, it's vital for cellular energy metabolism. It's a key player in something called the Malate-Aspartate Shuttle.
The white shuttle.
The Malate-Aspartate Shuttle. It's basically a mechanism that helps transport energy components efficiently into the mitochondria, the powerhouses of the cell, particularly in tissues that need a lot of energy, like muscle, liver, and heart. And second, aspartate is involved in the urea cycle. That's the body's main way of getting rid of ammonia, which is a toxic byproduct that increases during exercise.
Okay. So it's right at the intersection of energy supply and waste removal, 2 things that are obviously heavily stressed during physical activity, especially endurance activity like walking, which was key for the BAI score.
Exactly. Its dominant role makes sense when you consider those functions. It acts almost like a central indicator light for how efficiently the body's engine is running and cleaning itself during activity.
A clear molecular gatekeeper, summarizing the whole metabolic situation.
So identifying aspartate as the key marker is a massive step. But, science doesn't stop there. Finding the what is great, but the real understanding comes from figuring out the how and the why.
Right. We know active people have different aspartate levels.
Yeah.
But why? What's the underlying machinery controlling this? Correlation doesn't tell you the mechanism.
Precisely. We need to move from just looking at the levels of metabolites like a static snapshot to understanding the dynamics, the system that regulates them.
How do you even do that? How do you see the system in action?
That's where they used another really novel tool. It's a data-driven modeling approach called CovRecon. Stands for Covariance-Based Reconstruction. It uses something called an inverse differential Jacobian approach.
Okay, CovRecon. Inverse differential Jacobian. Let's stick to plain English if we can. If metabolism is like a city's road network and metabolites are the cars, what is CovRecon looking at?
That's a good analogy. CovRecon isn't just counting the cars, the metabolites. It's looking at the traffic flow patterns and, more importantly, the state of the infrastructure itself. The traffic lights, the intersections, the bridges, these are like the enzymes in our body controlling the flow.
Enzymes control the reactions, right?
Exactly. So CovRecon uses the data to estimate how the connections between these enzymes, how the network structure itself changes between the active group and the less active group. It reconstructs the network and pinpoints which enzymatic links are being most actively, let's say, rewired or adjusted by physical activity.
So it's looking for the dynamic changes in the control system, not just the outcome. Which parts of the metabolic highway are being upgraded or managed differently in active people?
That's a great way to put it. It's looking for signs of active network reorganization. And this network analysis pointed very clearly to 2 well-known enzymes as being the central hubs controlling this aspartate network.
Which enzymes?
Aspartate aminotransferase, or AST, and alanine aminotransferase, or ALT.
Wait a minute, AST and ALT, those sound really familiar. Aren't those the standard liver enzymes they check in routine blood tests? Like for liver health?
Exactly. That's what makes this finding so elegant. They are indeed routinely measured in liver function panels. But here, they didn't pop up as indicators of liver damage. Instead, they emerged as key indicators of how physical activity dynamically reshapes metabolism, particularly involving the interplay between the liver and muscle systems.
So the very same enzymes we check for liver problems are also acting as regulators for this key fitness marker, aspartate, showing how activity changes the system.
Yes. It highlights this deep connection between liver function, muscle activity, and overall metabolic state reflected by aspartate.
Did they have proof that activity actually changed the behavior or the responsiveness of these AST and ALT enzymes?
They did, and this is a crucial validation step. They looked at the routine blood tests taken from these participants over the 6-month study period, and they found that the levels of AST and ALT fluctuated more dramatically in the active group compared to the less active group.
More dramatically? How so?
Specifically in the active individuals, AST and ALT levels decreased significantly more in the first 3 months of the observation period, and then they increased significantly more in the subsequent 3 months compared to the less active group.
That's fascinating. So it's not that their levels were just higher or lower overall, but that their system was more flexible, more responsive to changes over time.
Exactly. That's what the researchers concluded. This dynamic fluctuation points towards active individuals having greater metabolic plasticity and flexibility in these key enzymatic systems in the liver and muscle. As one researcher put it, physical activity is literally rewiring our metabolism at the molecular level.
And this isn't a sign of damage, it's a sign of a healthy adaptive system that can ramp up and down more effectively.
That seems to be the case. It's an adaptive reorganization, making the system more responsive.
Okay, so let's follow this chain. We've got aspartate as the key fitness marker. We've got AST and ALT plasticity as the dynamic engine regulating it, connecting liver and muscle. Now, how does this potentially link to the brain? That was mentioned earlier.
Right. This is where aspartate's dual role becomes really important. We already talked about its role in energy metabolism, that Malate-Aspartate shuttle, vital for muscle, heart, liver, and its role in the urea cycle for clearing ammonia, which is critical during exertion.
So physical activity really puts a demand on those core bodily functions of aspartate.
It does. But aspartate isn't just working in the muscles and liver, it also plays a critical role in the brain.
As a neurotransmitter.
It acts as a precursor for neurotransmitters, yes.
Yeah.
And specifically, it activates a very important type of receptor called the N-methyl-D-aspartate receptor, or NMDA receptor.
NMDA receptors. Those are involved in learning and memory, aren't they?
Absolutely fundamental. NMDA receptors are crucial for synaptic plasticity, the ability of brain connections to strengthen or weaken, which is the cellular basis for learning, memory formation, and generally maintaining healthy neuron function.
Okay, so this is the bridge. Physical activity drives changes in aspartate levels and the flexibility of its regulating enzymes, AST, in the body. And aspartate itself is needed for critical brain functions like learning and memory via NMDA receptors.
That's the proposed molecular bridge. The study shows physical activity leads to these measurable metabolic shifts rooted in pathways involving aspartate and AST. And these very same pathways have direct implications for brain health and resilience.
So the suggestion is that the benefits of exercise for the brain might actually flow through this specific metabolic channel.
That's the compelling implication. To strengthen this idea, we need to look at external evidence. What do other larger studies tell us about AST, ALT, aspartate, and cognitive health?
Right. Does the wider scientific literature support this connection?
It does, interestingly. There are independent large-scale studies that have looked at people over long periods, and they've found that having lower levels of AST and ALT in midlife, or having an elevated ratio of AST to ALT, is associated with an increased risk of developing Alzheimer's disease or age-related cognitive decline years, even decades later.
Wow. So low or imbalanced activity of these same enzymes decades earlier might predict future cognitive problems.
That fits perfectly with the idea that dynamic flexibility of these enzymes, as seen in the active group, is protective.
It aligns very well. It suggests that having a responsive, flexible AST system might be neuroprotective, while a sluggish or dysregulated system could be a risk factor.
And what about aspartate levels directly in the brain in conditions like dementia?
There's evidence there, too. Studies have found that in patients with dementia, the levels of aspartate in the brain are often dysregulated. This is thought to reflect problems with neurotransmission and the overall health of neurons. Okay, so pulling it all together, this study identifies a specific metabolic signature of fitness: high aspartate, flexible AST, driven by physical activity. Other research links poor function in this exact system to higher dementia risk and finds aspartate dysregulation in dementia itself.
Exactly. So the overall implication is that these metabolic shifts from exercise, the ones we can now measure via aspartate and AST dynamics, might be a key mechanism through which physical activity helps protect the brain. It offers a plausible, measurable, amino acid-based pathway linking movement to cognitive resilience.
This really moves the conversation forward, doesn't it? It's not just exercise is good for the brain.
Yeah.
It's potentially exercise maintains the flexibility of the AST system, ensuring proper aspartate handling, which supports NMDA receptor function crucial for cognition. We're getting down to the biochemical nuts and bolts.
Absolutely. But as with any exciting finding, we need to be careful and consider the nuances and limitations. Science progresses by building evidence carefully.
Good point. So let's talk nuances. One thing we mentioned was the type of exercise. The study participants did resistance training, but the main fitness signal, the BAI, was dominated by walking distance and endurance measure.
Right, and this is actually quite important. The paper itself cites other research suggesting that the specific metabolites that change after resistance exercise can be quite different from those that change after endurance exercise.
So lifting weights might change your blood chemistry differently than going for a long walk.
Potentially, yes. And significantly, endurance activity has often been shown in other studies to have perhaps a stronger link to some anti-aging effects compared to resistance training alone. So the fact that walking distance was the key driver of the BAI, which then correlated strongly with the aspartate signature, kind of reinforces the idea that it's these endurance-related metabolic adaptations that are being picked up most strongly here.
That makes sense. And what about those AST and ALT enzymes again? Since they can be markers of liver damage, we need to be absolutely sure the fluctuations seen in the active group were healthy, right?
Definitely. And the researchers addressed this. They specifically looked at the ratio of AST to ALT over the 6 months. In cases of liver or muscle damage, this ratio often changes significantly and stays changed.
But that didn't happen here.
No. They found that while the absolute levels of AST and ALT fluctuated more in the active group, the AST ratio did not significantly change over time compared to the less active group. This strongly suggests the fluctuations were due to the normal metabolic demands of exercise temporary adaptive responses rather than any underlying pathology. It confirms the plasticity is likely a healthy sign.
Okay, that's reassuring.
Yeah.
Now, let's talk limitations. The study had 263 people, mostly elderly women in Vienna. How far can we generalize these findings?
That's a standard limitation in many studies. The authors are clear that while the core metabolic pathways they identified, like aspartate metabolism, are fundamental and universal, the exact numbers, the precise predictive accuracy, the specific importance of each metabolite might look a bit different if you studied, say, younger men or people from different ethnic backgrounds or with different baseline health statuses.
So the principle likely holds, but the specifics need broader testing in diverse populations.
Exactly. Replication and validation in larger, more varied cohorts is always a necessary next step.
And the big one, the link to dementia. You stressed this before, but let's reiterate that connection is still hypothetical based on this study alone, correct?
Absolutely critical point. The authors themselves state this very clearly. Their study focused only on physical performance and the corresponding metabolomics. They did not measure cognitive function or track dementia outcomes in these participants.
So they've identified a plausible biological mechanism, this aspartate AST pathway, that could link activity to brain health, based on other research. But they haven't proven that link directly in this work.
Correct. They've built the molecular bridge, but future studies need to actually test traffic going across it, studies that measure physical activity, these metabolic markers, and track cognitive changes over time in the same individuals. The potential is there, but the proof requires dedicated research.
What about the AI models? 91% accuracy is great, but were there any weaknesses?
They did mention one thing. When they tested the models using smaller subsets of their own data during the validation process, the performance, while still good, did dip slightly.
What does that suggest?
It usually suggests that the models, while robust, could likely become even more accurate and stable with more data. Feeding these sophisticated algorithms larger and more diverse data sets often helps them learn the underlying patterns even better and become more generalizable.
So bigger data is likely to make the predictions even sharper. What are the main next steps for this line of research then? Where does it go from here?
Well, two main directions seem clear. First, as we just discussed, scaling up and integrating, doing larger studies that combine everything. The physical measurements, the detailed metabolomics using tools like CoveRecon, and longitudinal tracking of cognitive health. That's needed to definitively test the liver-brain axis hypothesis.
And the second direction?
Refining the tools themselves, particularly CoveRecon. The researchers mentioned wanting to enhance it, for example, by incorporating more detailed information about how enzymes are regulated and the speeds of different reactions. This would allow for an even more precise reconstruction of how these metabolic networks adapt to activity and aging, building a higher resolution map, essentially. #tag outro.
This has been a really fascinating deep dive, tracing the path from walking speed all the way down to individual enzymes and their flexibility. Let's try to boil down the key takeaways from this study on active aging.
Okay. I think the first major takeaway is that your physical fitness, especially in aging, leaves a distinct molecular fingerprint in your blood. They showed it can be predicted with a really high accuracy, over 91%, using just 8 key metabolites, and the standout marker is aspartate.
Second takeaway, this molecular fitness signature seems most strongly linked to endurance. Walking distance was the key physical measure driving the correlation, underlining the importance of sustained activity for this particular metabolic profile.
Third, active aging isn't just about having better levels of certain molecules, it's about metabolic flexibility. The study showed greater plasticity, more dynamic fluctuation in those key enzymes AST and ALT in the liver and muscle systems of active individuals. That flexibility seems to be a hallmark of a healthy adaptive response.
And finally, the fourth point. This research maps out a specific, plausible molecular pathway, the aspartate and AST axis, that directly connects physical activity to brain-relevant processes. It provides a testable mechanism for how exercise might protect cognitive function, bridging the liver, muscle, and brain.
And the study's conclusion that physical activity rewires our metabolism at the molecular level really brings it home. It shifts the perspective, doesn't it?
It really does.
It suggests that looking forward, we might be able to track how well you're aging, not just by counting your steps or measuring your strength, but by directly measuring the dynamic molecular network that your activity is building and maintaining inside you.
That moves us beyond just general advice like be active towards something much more personalized and quantifiable.
Exactly. Which leads to, I think, a provocative final thought for you, our listener. Knowing now that things like aspartate metabolism and the flexibility of enzymes like AST and ALT seem to be these key molecular readouts of fitness and potentially resilience, what measurable impact is your current routine, your level of physical activity, actually having on this deep metabolic machinery? This research is exciting because it's starting to give us the specific biomarkers like aspartate, like AST dynamics, that we could potentially use to ask that question precisely. To bridge the gap between just doing exercise and knowing if that exercise is optimally tuning your metabolism for long-term health, including the health of your brain, we now have clues about which signals to actually look for.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.